Welcome to Session 4 of the APRISE AI Skills and Generative AI Training course, AI for Literature Review. This course was produced by Telewander APS for the APRISE project through human expertise supported by artificial intelligence tools. All educational content, visual materials, and final outputs were reviewed and approved by the Telewander team. In this session, we will explore how AI can make literature review work more efficient while keeping researchers responsible for evidence quality, interpretation, and final decisions. By the end of this session, you should be able to identify literature review tasks that AI can support, create more effective search strategies, use AI to organize and compare evidence, recognize common risks, such as fabricated references and outdated information, and apply a practical verification workflow. The aim is not to automate scientific judgment. It is to use AI as a structured assistant while preserving traceability, critical thinking, and accountability at every stage of the review. Literature review is especially important in alternative proteins because the field draws evidence from many disciplines. A single research question may involve food science, biotechnology, nutrition, consumer behavior, environmental assessment, economics, and regulation. The evidence may also change quickly as technologies develop and new products reach the market. A good review helps us understand what is known, where findings disagree, which methods were used, and what gaps remain. AI can help manage this complexity, but the quality of the review still depends on the researcher's question, inclusion criteria, and judgment. AI can support many repetitive and language-intensive tasks. It can help generate keywords, expand synonyms, draft Boolean search strings, summarize text that you provide, extract recurring themes, suggest comparison categories, and reorganize notes into tables. It can also explain technical concepts for different audiences and help identify claims that require verification. These uses can save time, especially during early exploration. However, an AI output is not evidence. It is a working draft that must remain connected to the original sources. AI cannot reliably guarantee that a paper exists, that a citation is accurate, or that a study is methodologically strong. It may invent authors, titles, journals, or DOI numbers. It may overlook limitations, confuse correlation with causation, misread statistical results, or present outdated regulatory information as current. It can also produce confident conclusions that are stronger than the underlying evidence. For these reasons, never treat an AI-generated reference list or summary as verified. Open the original source, confirm its identity, and read the relevant sections yourself before using the information. This workflow shows where AI can assist without replacing the researcher. First, define a focused question. Second, establish the scope and eligibility criteria. Third, create search concepts and synonyms. Fourth, search appropriate databases and official sources. Fifth, remove duplicates and screen records. Sixth, retrieve and read the full texts. Seventh, extract data using consistent fields. Eighth, compare and map the evidence. Ninth, verify claims, references, and interpretations. Finally, synthesize the findings and document the process. At every step, preserve the link between your notes and the original source. Human review is not a final add-on. It is part of the entire workflow. We will use one example throughout the session: consumer acceptance of fermentation-derived proteins in Europe. This topic is broad enough to involve product terminology, consumer attitudes, sensory expectations, perceived naturalness, safety, sustainability, price, and regulatory context. It is also narrow enough to demonstrate a structured search. A useful review question might ask which factors influence European consumers' willingness to try or purchase foods containing fermentation-derived proteins, and how those factors vary across products, populations, and study designs. AI can help us move from a research question to a search plan. The prompt on this slide gives the tool a clear role, a specific topic, and a defined output: concepts, synonyms, spelling variants, and a Boolean search string. We should also state which database we intend to use, because syntax and indexing differ. The first output is only a draft. Review every term, remove irrelevant suggestions, and add terminology used by the field. A search strategy should be transparent enough for another researcher to understand and reproduce. For our example, the search can be organized into 3 concept groups. The first describes the technology or ingredient, such as precision fermentation, biomass fermentation, microbial protein, or fermentation-derived protein. The second describes the outcome, including consumer acceptance, willingness to try, purchase intention, perception, or attitude. The third defines the context, such as Europe, the European Union, or individual countries. Within each group, synonyms are joined with or. The concept groups are then joined with and. This example search string applies that logic. Parentheses keep related synonyms together. Or broadens the search by accepting any term inside a concept group. And narrows the search by requiring the main concepts to appear together. Quotation marks can preserve exact phrases where the database supports them. Before launching a large search, test the string with a small set of known relevant papers. If those papers do not appear, revise the terminology, fields, or filters, rather than assuming the database has no evidence. No single search tool covers every type of evidence. Bibliographic databases are useful for peer-reviewed research. Specialist repositories may cover particular disciplines. Citation tools can help identify related studies. Official institutions, such as the European Commission, EFSA, and FAO, provide regulatory and policy information. Industry reports may offer market context, but their methods and interests must be examined. The photograph reminds us that source selection benefits from discussion. Agree in advance which sources are appropriate, and record where, when, and how each search was performed. AI can summarize an article more reliably when you provide the verified text or your own extracted notes. Ask for separate fields: research aim, method, sample, intervention or exposure, main findings, limitations, and relevance to your review question. Also ask the tool to distinguish what the authors reported from any interpretation it proposes. Do not request a summary from the title alone, and do not assume the model has access to the paper. After receiving the summary, compare it with the abstract, methods, results, and discussion. If a claim matters to your conclusion, verify it directly in the original article. A consistent template makes summaries comparable. Start with the complete citation and a stable identifier, such as a DOI, PubMed record, or official URL. Record the research question, study design, population or material, sample size, methods, key outcomes, and limitations. Add a field for relevance to your review, and another for claims that require checking. The citation should be copied from the verified source or a trusted reference manager, not generated from memory by the AI. This structure also creates an audit trail. Every later statement can be traced back to a specific record and passage. AI can help extract or restate research questions from verified abstracts and full texts. This is useful when authors phrase their aims differently. Ask the tool to quote or closely paraphrase the stated objective, identify the population, intervention or phenomenon, comparison, and outcome, and indicate where the information appears. Then review the extraction yourself. Similar language does not always mean that two studies answer the same question. Differences in product, geography, population, and measurement method can change what the findings mean. Comparison tables are one of the most useful AI-assisted outputs. Once the data have been verified, AI can reorganize them into common columns, such as study design, sample, product type, country, outcome measure, main result, and limitation. This helps reveal patterns and differences. But a tidy table can create a false sense of equivalence. A small qualitative study and a large representative survey should not be treated as equally strong simply because they occupy adjacent rows. Preserve methodological differences and include a source identifier in every row. Evidence mapping provides a structured overview of what has been studied. It can show which technologies, outcomes, populations, and regions receive attention, and where evidence is sparse. AI can help classify verified records into predefined categories and suggest additional categories when patterns emerge. The categories still need human review, especially when a study fits more than one area. An evidence map describes the distribution of available evidence. By itself, it does not establish study quality or prove which option is best. This matrix turns the review into a visible pattern. Rows may represent technologies or product categories, while columns represent outcomes, stakeholder groups, or regions. Each cell can record the number and type of studies, with links back to the source records. Dense cells show where research is concentrated. Empty or thin cells suggest possible gaps, although a gap in the matrix may also reflect limitations in the search. The matrix is a navigation tool. It helps researchers decide where deeper synthesis is needed and which stakeholders may be affected by missing evidence. AI can also translate complex concepts into language suited to different audiences. You might ask for a technical explanation for researchers, a plain language version for consumers, or a short briefing for decision makers. Always specify the audience and the required level of detail. Then check that simplification has not changed the meaning. The next 4 examples: extrusion, precision fermentation, cell culture media, and life cycle assessment show why accurate context matters. Extrusion combines ingredients under controlled heat, pressure, moisture, and mechanical shear. In alternative protein products, it can help transform protein-rich materials into structures with fibrous or meat-like texture. The result depends on the raw materials, equipment, and processing conditions. An AI explanation should therefore avoid claiming that extrusion produces one universal outcome. It can help clarify the process, but technical details must be checked against reliable food engineering sources and the specific product context. Precision fermentation uses microorganisms as production hosts for specific target molecules, such as proteins, fats, enzymes, or other ingredients. The organism is cultivated under controlled conditions, and the target product is then recovered and purified as required. This is different from saying that every fermentation-derived food follows the same process or regulatory pathway. When AI summarizes this area, verify the organism, product, intended use, production method, and jurisdiction before making technical or regulatory claims. Cell culture media provide cells with nutrients and environmental conditions needed for growth and function. They may contain amino acids, sugars, salts, vitamins, growth factors, and other components. Requirements differ by cell type, process stage, and intended product. AI can help organize information about media components, but it may merge details from incompatible systems. Check the original protocol, species, cell line, concentration, experimental conditions, and date before applying any summary. Life cycle assessment, or LCA, evaluates environmental impacts across a defined product system. Results depend on choices such as system boundaries, functional unit, allocation method, energy assumptions, geography, and data quality. Two studies may report different impacts without directly contradicting each other because their assumptions differ. AI can help extract these parameters into a table, but it should not compare headline numbers until the underlying methods are aligned. Always interpret an LCA result within its stated scope. One of the most serious risks is the fabricated reference. AI may combine a real author, a plausible title, a journal name, and a convincing DOI into a citation that does not exist. It may also attach an incorrect finding to a real paper. To verify a reference, search the exact title in a trusted bibliographic database. Confirm the author's, year, journal, and identifier. Open the original record and check that the claimed result appears in the paper. If the reference cannot be located, do not cite it. Ask the AI to work only from a reference list you have already verified, and keep the source link beside every extracted claim. AI outputs can also be outdated, particularly in fast-moving scientific and regulatory areas. A general statement such as, fermentation-derived foods follow the same approval pathway everywhere in Europe is too broad. Regulatory status depends on the legal definition, product characteristics, production process, intended use, and jurisdiction. For EU novel food questions, consult current European Commission and EFSA information and record the access date. Use qualified language, identify uncertainty, and avoid extending a rule from one product category to another without evidence. AI can accurately repeat individual sentences while still misinterpreting the study as a whole. It may turn an association into a causal claim, treat statistical significance as practical importance, ignore confidence intervals, or overlook a non-representative sample. It may also confuse an author's hypothesis with a finding. When reviewing a summary, ask, what was actually measured? Compared with what? In which population? Over what period? With what uncertainty? Then read the relevant tables, figures, and limitations in the original paper. Generative AI often prefers a clear answer, even when the evidence is mixed. It may say that a technology is sustainable, safe, or accepted when the reviewed studies only suggest a possible trend under limited conditions. Replace absolute wording with language calibrated to the evidence. The available studies suggest, within the assessed scenarios, or evidence remains limited. Confidence should come from transparent methods and converging evidence, not from the fluency of the sentence. Use this 5-step verification workflow for any important AI-assisted output. First, identify the exact claim and the source it supposedly relies on. Second, confirm that the source exists and that its bibliographic details are correct. Third, open the original source and locate the supporting passage, table, or figure. Fourth, check context, study design, population, methods, limitations, date, and jurisdiction. Fifth, record the result of the check and revise or remove unsupported wording. For high-impact claims, use a second reviewer or an independent authoritative source. This process converts an attractive draft into a traceable research output. Now apply the workflow to a topic relevant to your work. Define one focused review question. Ask an AI tool to suggest search concepts and a draft Boolean string. Select a small set of sources that you can verify. Create a comparison table with consistent fields and identify at least 3 claims that require checking. Finally, record how you verified one reference and one substantive claim. The activity should take about 15 minutes. Pause the course if you want to complete it now, or save the task for facilitated discussion. An effective output is compact but traceable. It may include the review question, the final search string, a table of verified studies, an evidence map, and a list of claims requiring follow-up. The analytics image reinforces an important point. Organizing information is not the same as interpreting it. Researchers still decide whether categories make sense, whether studies are comparable, and whether the evidence supports a conclusion. Save the prompt, date, tool, source list, and human corrections as part of the research record. A stakeholder table helps connect literature findings to real decisions. Researchers may focus on evidence gaps and methods. Product developers may need information about functionality, cost, and scale-up. Regulators require precise product definitions and current legal evidence. Consumers may care about taste, price, safety, naturalness, and sustainability. Farmers, ingredient suppliers, and civil society groups may have different concerns. AI can help organize these perspectives, but it should not invent stakeholder views. Use published evidence or direct engagement. Some claims should trigger immediate verification. Does a cited paper actually exist? Does the study support the stated conclusion? Is the information current? Does the result apply to the same product, population, and jurisdiction? Are environmental comparisons based on compatible system boundaries? Is a consumer preference result representative beyond the sample studied? Turning these claims into verification questions prevents plausible language from becoming unsupported evidence. The more consequential the claim, the stronger and more independent the verification should be. The key message is that AI can accelerate literature review tasks, but it cannot take responsibility for the review. Use it to expand search terms, structure extracted information, compare verified studies, and communicate complex concepts. Do not rely on it to confirm references, evaluate study quality, or make final scientific and regulatory judgments. Keep every important claim traceable to an original source. Document your process and use calibrated language. Efficiency is valuable only when evidence quality is preserved. Before moving on, consider 3 questions. Which part of your current literature review process could AI support safely? Which task should remain under direct human control? And what evidence would you need before trusting an AI-assisted conclusion? Your answers may differ by project, discipline, and risk level. The goal is not maximum automation. It is a deliberate division of labor, in which AI supports speed and structure, while people retain judgment and accountability. Used well, AI can help researchers ask better questions, build clearer searches, organize complex evidence, and communicate findings more effectively. Used carelessly, it can amplify errors with convincing language. The difference is the workflow: verified sources, transparent prompts, consistent extraction, cautious interpretation, and human review. Treat AI as a capable assistant, not as an authority. The researcher remains responsible for what enters the review, how evidence is interpreted, and what is ultimately communicated. This session draws on established guidance for transparent literature searching, systematic review, and responsible use of generative AI. The reference slide includes PRISMA guidance, the Cochrane Handbook, UNESCO guidance on generative AI, research on fabricated references, and official European sources for novel food information. Use these as starting points, and always consult the latest version of an official or methodological source. The complete citation details remain on screen for your records. You have completed Session 4. Use the QR code or the link on screen to open the assessment. It includes scored multiple-choice questions, followed by unscored reflection questions, organized into 3 sections. Take time to consider how you will verify references and claims in your own work. In the next session, we will explore how AI can support alternative protein product development.